So Agtc, we are a publicly traded company, so here's the forward-looking legal statements. As a highlight for our company, we are focused on orphan ophthalmology indications, and we are a gene therapy company using the adeno-associated virus. One thing I really like to drive home is the years of experience that we have in this area. Our founders are leaders in their field. They've got decades of experience. The company has been in existence for almost 15 years, and we really value our deep understanding of the virus that we're using and how to construct it to match specific phenotypes of the indications we're working on. These are our lead products. As you can see, we have a very broad pipeline, and I won't go into too much detail, but the takeaway here is that we have 4 clinical trials ongoing and 4 different indications, and we'll have a 5th one in the clinic in 2018. So I want to talk a little bit more about that comprehensive platform and that deep understanding of viral vectors. I think all of us in this room know a lot about viral vectors, but sometimes when you talk to potential partners or investors, they kind of think of the vector as just a singular one pony kind of thing. And what I like to make sure people understand is that to devise these vectors, you have to understand the capsid or the serotype, you have to understand the promoter that drives gene expression. You have to specifically engineer the gene. You got to get your formulation right, especially in the eye. You've got to think about manufacturing and then that physical delivery. And all of these parts are equally important. All of these parts need to work well together and be specifically designed for where it is you're trying to deliver. And so looking back at our pipeline again and using that idea of what is the key platform component, I'm just going to point out a couple of examples. XLRS is an orphan indication in the eye where there's a structural protein that's missing. So the retina, the back of the eye, is very fragile. So the most important thing for us was to use the least invasive delivery we could. And so we wanted to do an intravitreal injection, and then we designed all of the rest of the product around that component. For achromatopsia, it's a different story. It's still orphan ophthalmology, but what's missing here is a cell membrane protein that opens a channel. And it was very important that we didn't want to open that channel in any other cell type except the cell that needed that channel. And so the part of the vector that was most important was the promoter. It's a cell-specific promoter. So we designed and patented a cone-specific promoter, a primate cone-specific promoter, and then we designed the rest of the product around that specific indication. So I use those as examples of how we think about product development and the attention we play to the science and the technology and designing all of these very specifically. We are not anywhere near a one-size-fits-all organization. We're designing each individual component quite carefully. So why ophthalmology? I'm not going to spend too much time on this. I think that this audience understands that in gene therapy, ophthalmology is a very attractive area. It's a significant unmet need. Before gene therapy, there weren't really people working in the orphan space in ophthalmology, and there's a significant unmet need across the literally hundreds of different genetic deficiencies that cause vision loss. So this is the eye. Again, I'll use this slide to point out how specific you have to be in ophthalmology. Depending on which side you are, on your right-hand side is a blow-up of the retina. So the retina is this very thin part of the eye at the back, but it is highly differentiated. And each individual cell looks different, as you can see from this cartoon, but also has a different function. And depending on which genetic disease you're working on, you have to be able to get to that specific cell type. So again, this deep level of understanding, not just of the vector, but of the organ we're working in, is very important to our forward progress. So I'm not going to spend too much time on each individual program. I have quite a bit of slides here on each individual program. I'm still going to try and use this to talk about differentiation of in orphan ophthalmology. So X-linked retinoschisis, or XLRS, I've already mentioned this is a structural protein that's missing. One of the things that is important to us is a fairly large patient population, about 35,000 patients in the US and Europe. That's an important characteristic to us when we're selecting indications, that they have a substantial patient population. Also, another feature of this indication is that for the patient, their disease is stable over long periods of time, decades even. And that means that for this indication, we don't necessarily have to kind of catch them when they're young. We have an opportunity to have an effect on these patients and treat a larger percentage of the prevalent population, and that was an important selection criteria for us. So this slide you'll get pretty tired of by the end of my presentation. One of the things about having multiple trials in the clinic in the same kind of indication, all orphan ophthalmology, is that our clinical trial design starts to look repetitive, which we can leverage the knowledge and how we're developing protocols. So this is a standard dose escalation portion, and then there's a second half of the trial, which is expansion of the number of patients at the maximum tolerated dose. In XLRS, we have completed that dose escalation portion and are now dosing patients at the highest dose, which was into the expansion cohort. So what we've announced about that dose escalation phase is it is complete. The safety was that it was generally well tolerated with only mild to moderate inflammation that either resolved spontaneously or was well controlled and resolved with topical or oral corticosteroids. We are continuing to gather the data from that dose escalation phase, but we've advanced into 2 additional cohorts. We're now treating kids at the mid-dose group. We thought in an abundance of caution, sometimes it's been shown in the past that children have an advanced or an accelerated immune response, so we wanted to go down dosing in kids, and then at the highest dose we're dosing in the expansion cohort, and we've already dosed 5 patients in that cohort. Achromatopsia, again, I've mentioned this before. This is a cone-specific disease. It's a channel membrane, a cell membrane protein that forms a channel. These patients are all legally blind and extremely light sensitive. There's no way they'd be standing up here with these 2 lights shining in their eyes. And again, it's a pretty substantial patient population, about 28,000 patients in the US and Europe. So again, the clinical trial design should look familiar, dose escalation and then an expansion cohort. And we've now dosed 4 out of 5 of the total patients in the low dose. And as soon as we dose that 5th patient and meet with our Data Safety and Monitoring Board, we'll advance to the next cohort. We have 3 active sites with a 5th doing some really pretty cool optical testing where we're able to actually visualize individual cones in the patient's eyes. So this is a nice tool for us to be able to use as we advance this program. The next program is X-linked retinitis pigmentosa, or XLRP. Now, RP, or retinitis pigmentosa, is usually the first indication I talk about that people go, oh yeah, I've heard of that one. It is the largest of the orphan diseases. However, RP in general is caused by mutations in as many as 100 different genes, but XLRP is caused primarily by mutations in one gene, and it's about 20,000 patients in the US and Europe. So now you've heard me say 3 times that there are 20, 30, 40,000 patients in the indications we're working on. Again, that was an important consideration for us. XLRP, the IND was filed this summer, and we recently were able to announce that that has cleared, and we're now undergoing all the site initiation visits at the different clinical sites to bring this forward. It's going to be the exact same dose escalation and then an expansion cohort. And I think that I'll just sit this here a minute to show what we think is the power of what we're doing, in that, as you can see, we have diseases that are very large patient populations. They're single gene. We've designed the vectors specifically. All the vectors for these different indications are different from each other and specifically designed for the cell type and the phenotype of the disease. We are able to leverage this dose escalation and expansion, and we have sites throughout the United States that are academic referral centers and are specialists in these retinal diseases, and our sites are overlapping. So at many sites where they've already initiated the XLRS trial, they've already initiated the achromatopsia trial, so being able to initiate and bring forward XLRP is we're utilizing what we've learned to be able to advance these programs more and more effectively as we move forward. I think the other thing to point out is that while many people know that they have a genetic form of blindness, up until very recently there was nothing that could be done. So they were basically told, you have a genetic form of blindness, there's nothing we can do for you, and many of them were not genotyped. So one important thing that we've done is bring on patient advocacy and make very deep relationships with many of the patient advocacy groups across the country and the world, and bring them in and get the word out that now these patients should get genotyped. And in fact, we recently funded the Foundation Fighting Blindness so that they can offer genotyping free to the patients that join their patient registry. And I think this is going to be very important for us. That registry is going to be open to the entire industry, and so I think it can be very important for others. But I think this is a component as we get more and more sophisticated moving our products forward that we're going to have to pay attention to, that patients have to know that there's something available for them potentially now. They have to know that there's an opportunity to get genotyped. Here we go. So Bionic Sight, this is a collaboration we have to work in optogenetics, and I'm not going to talk too much about this. This is a program that's going to go into the clinic next year. We have a few little bells and whistles in the optogenetic space, including a non-implanted wearable device and a channel rhodopsin, or a light-sensitive protein that is sensitive across a much wider range of light intensity, which we think gives our program an advantage in the marketplace. I'm not going to talk about otology today, but we feel there's a lot of similarities between the eye and the ear. Let's just talk about photoreceptors turning light into electrical signals. Hair cells turn sound waves into electrical signals. So we think we can make a lot of progress in the ear based on our knowledge of the eye. We have a very experienced team. This is the typical kind of logo scattershot, but you can see that our team has a lot of experience both in ophthalmology and gene and cell therapy. And the thing that I want to kind of end the presentation on is manufacturing. You heard the panel before talk about manufacturing. This is something we've paid a lot of attention to from the very beginning of the company. And I think we have now made clinical trial material that has not been put on clinical hold by the FDA or the EMA for 7 different indications. There's very few organizations out there that have made 7 different vectors, gotten it through regulatory review, and used that material in the clinic. So we feel very secure about our ability to manufacture, about the robustness of it and the repeatability of it, and our ability to move forward into pivotal and commercialization. On top of that, we feel that in some respects, gene therapy has a little bit lagged. They've been focused so much on that manufacturing issue that they haven't made the advances in characterization that the FDA is going to want in commercialization. And so we've spent a great deal of time over the last 2 years switching over from what could be called kind of academic qualitative kind of testing to more industrialized, quantitative, robust characterization tests, and those are things that we're now moving forward into operations. So again, my CFO would be very mad if I didn't spend some time on this slide. We do have $138 million on board, which is enough funds for us to complete those clinical trials I talked about, as well as advance all of our research programs I didn't have a chance to talk about for at least the next 2 years. So broad pipeline, very good balance sheet, focus on the science and the technology. So thank you for your time, and have a good rest of your conference.